Okay, questions? None? Okay. I think we'll be able to finish up our kind of run through the cell structure of bacteria today and get started on a new topic on Tuesday, which would be nice because what we're going to start on Tuesday deals with classification bacteria and identification of bacteria, and that'll introduce you to Birdie's manual. And that's appropriate because next week in lab on Wednesday, you're going to get your unknown. Okay? So we'll start working on that. So we are working on the cell envelope portion of the bacterial cell. And we spent a lot of time talking about the cell wall. And at the end of the period, some of the exceptions to the standard. If you're a bacteria, you have peptidoglycan cell wall structure. The other part of the cell envelope we're just going to briefly mention is the cell membrane. And the reason that we're going to just briefly mention this is because you should be well versed from cell biology on basic membrane structure. Now, granted, these are prokaryotic cells as opposed to the eukaryotic cells we talked about in cell biology. But still, the basic organization of the cell membrane is the same in both types of cells. Phospholipid bilayer, proteins, many of those proteins are involved in transport function in the bacterial cell membrane. What was one of the differences I said there was between prokaryotic cell membranes and eukaryotic cell membranes? What do eukaryotes have that prokaryotes generally don't have? Sterile molecules like cholesterol. And remember that those sterile molecules give some rigidity to the membrane. What is the exception to the rule of prokaryotic membranes not having sterols? Mycoplasms, because they lack what? Cell wall. And cell wall adds to the rigidity of the cell. And so if you don't have that cell wall, you need some compensating factor, and those sterile molecules help to give those mycoplasms a little bit of additional rigidity. Do prokaryotic cells, like bacteria, have the ability to carry out aerobic cellular respiration? Actually, they do. Okay? What did we need to synthesize ATP by chemiosmosis during aerobic cellular respiration in a eukaryotic cell? What powered the synthesis of the ATP? What was the source of energy?
Gradient.
Good. That hydrogen ion gradient? Okay. And the hydrogen ion gradient was set up across what membrane? The inner mitochondrial membrane, right? We had high concentration in that inner membrane space and low concentration in the matrix. So in the eukaryotic cells, we need that mitochondrial membrane, we need a mitochondria to make ATP by aerobic cellular respiration. What about prokaryotic cells? Do they have a mitochondria? Membrane-bound organelles in prokaryotes? Nope, no mitochondria. And yet they can carry out aerobic cellular respiration. Do they have to have a hydrogen ion gradient across the membrane? You bet they do. But they don't have a mitochondria. What do they use? They use their plasma membrane. They use their cell membrane, and therefore set up a gradient between the cytoplasm of the cell and the near external environment. So a little bit different strategy. But even the fact that they lack the internal cell organelles doesn't preclude them from making ATP by that efficient process of aerobic cellular respiration. They just use a different membrane system, the plasma membrane, to do that. Okay? One of the things you'll also see, not uncommonly, in prokaryotic cells is that the plasma membrane sometimes evaginates or protrudes into the cytoplasm of the cell. So this would just be an extension of the plasma membrane, kind of filling into the cytoplasm of the cell. This particular structure is called a mesosome. By looking at my drawing right here, what do you suppose the adaptation is of a mesosome? What's its purpose? Increasing surface area. Okay, so if some of these membrane-bound, if some of these cell processes that are associated with membranes are associated with the plasma membrane, increasing the amount of plasma membrane is going to be an adaptation for having more efficient enzymatic processes, more surface area, more enzymes. Okay? These mesosomes are also involved in cell division. Remember, bacterial cells divide by binary fission. And so during cell division, one of the things that happens is the DNA of the bacterial cell can attach to this mesosome, and this can help to facilitate separating it into the two halves of the cytoplasm as the cell divides. So it plays a role there. And we're also going to see that the mesosome can also play a role in the formation of endospores. They can kind of segment off a little part of the cytoplasm where the endospore takes place. And so we'll show you that in just a second. Remember that, according to the three-domain system, we have two major big groups of bacteria. We have the domain bacteria, and we have the domain archaea. Members of the domain bacteria have a cell membrane structure just like I've been talking about, pretty much like eukaryotes, lack of sterols, etc. But in the archaea bacteria, there are some fundamental differences in membrane structure compared to the domain bacteria. And one of them has to do with the phospholipids that are found in archaea and regular bacteria, and the way those phospholipids are joined together. Here's our typical phospholipid representation, showing a couple of unbranched fatty acids, and then our glycerol. Here's our phosphate that's part of the phospholipid. And these are hooked together by ester bonds. I don't know if you remember ester bonds from cell biology. Okay? So this would be the kind of phospholipids we typically find in eukaryotic cells and also in bacteria that are in the domain bacteria. In archaea, phospholipids are a little bit different. And they're joined together by a slightly different type of linkage that joins the glycerol to the fatty acids. They're joined together by an ether bond. You have to know the structure of those? No. But you should make that association of ether bonds with archaea bacteria and ester bonds with the other bacteria. So what's the significance? Well, this particular linkage, this ether linkage, is a little bit stronger, a little bit more stable, a little less likely to break. And why is that significant? Because a lot of those archaea bacteria are types of bacteria that are called extremophils. And these are bacteria that typically live in very hostile type environments. So having that little bit of extra strength in those phospholipid bonds can be important in those particular organisms maintaining their ability to live in those extreme environments. Not sure what happened to my... That's not good. Okay. Okay, let's move into the cytoplasm of the cell and talk about some cytoplasmic structures that we will find in bacteria. The one cytoplasmic structure that we're going to spend the most time discussing, and that will be quite significant for some of you in terms of identification of your unknown, are the endospores. What does endo mean? Inside? Okay, so these are structures that are formed within the cytoplasm of the cell. Typically, a single cell will only produce one endospore. So this is not a method of kind of reproduction. It's not like one cell can form all of these spores, each of which could give rise to an individual cell itself. One cell produces one endospore. That process of producing an endospore is called sporulation. It doesn't take that long for sporulation to happen, somewhere between 6 and 8 hours before a spore can form. Under the right conditions, one endospore can then germinate, and that's a faster process. That only takes an hour or two to form one cell. So what do you suppose might be the advantage of a single cell forming this structure? And when this single cell forms this structure, the cell that formed the endospore, which is called a vegetative cell, will die. It'll disintegrate. So now we've just got that endospore sitting there. But then, under proper conditions, that endospore can then germinate and give us that single cell back again. So this is not a method of increasing your numbers. What might this be an adaptation to? What would be the advantage of a cell making a spore, the cell dying? The spore eventually gives rise to a new cell.
Maybe some kind of repair mechanism?
Maybe some kind of repair mechanism, okay?
Basically fighting against antibiotics.
Okay, so something in the environment that could be a threat, like an antibiotic. There's a lot of potential threats to the environment. Antibiotics could be one. What about running out of food? What about it gets too hot? What about the environment becomes extremely dry, desiccation? Okay. So typically what's going to happen is there's going to be some environmental trigger, typically in the form of some type of stress, that's going to initiate that process of sporulation. And then if those conditions change and the environment becomes more favorable again, then the endospore can germinate and we've got our cell again. Okay? So it's a way basically to wait out bad environmental conditions. Endospores are remarkably resistant to any number of environmental insults, including high temperatures, lack of food, radiation, antibiotics potentially. how long can an endospore persist in that kind of dormant state? Because that's what we're going to see. Endospores have cytoplasm. They have nuclear material. They're basically a cell in waiting, waiting to germinate and give rise to a new actively dividing cell. How long can they lie there dormant? A long time. Okay? Years. Hundreds of years. Potentially thousands, or if you believe some accounts, millions of years. They have found endospores from fossilized bee crystals and fossilized salt crystals, millions of years old. So these are remarkably resistant structures that can hang around, and wait until the environmental conditions get better and then germinate again. Do all species produce endospores? No. One of the genera that is notorious for producing endospores is the genus Bacillus. Members of the genus Bacillus are gram-positive rods. You looked at some Bacillus in lab last week, those rods that were in chains. And not all bacteria produce them. Cocci, very rarely. The short gram-negative rods, very rarely. And so if you have a bacterium that is a gram-positive rod, and other data suggests that it is Bacillus, then endospore formation is going to be really an important tool in identifying species within that particular genus. Some of you will have Bacillus species as your unknown. And so endospores will be pretty critical for you. Here is a phase contrast microscopy preparation of living bacterial cells that contain endospores. And here you can see that the endospore is found at the end of the cell. It's that swollen area at the end of the cell. Here are some free endospores where cells have died, and the vegetative cell that gave rise to the endospore has already died and broken up and disintegrated. And here are some vegetative cells that have not yet given rise to endospores. They haven't produced them. This cell that gives rise to the endospore is referred to as the sporangia. Sporangia are the structures that produce the endospores. Some of you are familiar with that term from botany. We heard about sporangia. But here, the cell itself is the sporangia. That's going to be important because when you start identifying Bacillus species in Bergey's manual, one of the first things you will see in the key is the sporangia is swollen or the sporangia is not swollen. What does that refer to? That refers to whether this endospore inside of the cell, the sporangium, has distended the cell and made it look swollen or not. In addition to whether the cell is swollen or not, other things that are critical in terms of identifying different species of Bacillus is the location of the endospore within the cell. Is it at the end of the cell? In that case, it's said to be terminal. Sometimes it's in the middle of the cell. That's central. And probably most frustrating is in Bergey's manual, you oftentimes have to make a differentiation of size of the endospore. So one of the breakpoints in the dichotomous key is endospore is 0.9 microns or greater or less than 0.9 microns. Well, if you remember some of your measurements that you did, and those cocci were, about a micrometer in diameter, making that determination on endospore size can be kind of tricky. Okay? I'll talk more about that and what you do in that situation where, man, I'm not really sure if it's 0.9 or less. There are some other strategies we can use. This is just a colorized representation, a fluorescent antibody stained microscopic preparation of the cell, showing a vegetative cell. And here is an endospore that is forming in the terminal portion of the cell. Blue represents the DNA. So you can see here that some cytoplasm is being segmented off and surrounding the DNA of the cell, and this eventually will give rise to that highly resistant endospore. Here's the main chromosome of the cell, and here is the cytoplasm of the vegetative cell. Eventually this will disintegrate away, leaving our endospore. We're going to do a spore stain in class. This is a particular type of endospore stain called the Schaefer-Fulton method, and it'll describe the endospore stain in the lab handout when we get there in a couple of weeks. But basically this particular stain is a differential stain, and the cytoplasm of the vegetative cells stains red, and then the endospores stain a green color. So there's a technique you can use to differentially stain the endospores. Here we see a lot of free endospores that have been released from cells. One of the things that can be a little bit tricky when you're doing the endospore stain is finding an intact vegetative cell with an endospore inside of it, because that's the only way you can tell its cellular location, for example. One of the reasons that you get so many free endospores is that this particular staining technique involves some real harsh treatments to the cells, like boiling the stain by taking a Bunsen burner and turning it upside down and boiling the stain that you put on the surface of the slide to drive it into the endospore, because otherwise endospores are really hard to stain. So as you might imagine, boiling cells in stain destroys most of the cells. The endospore itself is a pretty complicated structure that consists mainly of 3 layers or 3 regions. So here is our vegetative cell. Here's the cell wall out here. Here's the cytoplasm. And then here is our endospore right here. The central region of the endospore is called the core. And the core contains cytoplasm, DNA, ribosomes, and then it's surrounded by a cell membrane. Basically, those are the requirements for being a cell, right? So this is kind of a little mini cell within the vegetative cell. Surrounding the core is a multi-layered structure called the cortex. And we'll talk about the cortex in just a minute, but there are some chemicals in the cortex that are responsible for the high degree of resistance that the endospore possesses. So we'll come back and refer to that in just a minute. And then there's an outer layer of the endospore called the exosporium. So exosporium, cortex, and core. So let's take a look at the core for just a second. In addition to the materials that I've listed being in the core, there's also water. I mean, cytoplasm is water, right? Do you remember what the typical cellular cytoplasmic concentration is of water? Is there a lot, a little? A lot. I mean, typically, 75, 80% of the cytoplasm is water. In the core, the cytoplasmic concentration of water is down in the 25% or less range. So the cytoplasm is very dehydrated. And that's intentional. One of the things that happens in that low water environment its metabolic reactions are slowed down tremendously. Well, the slower your metabolic rate, the less resources you require, the less food you require, for example. There's another advantage to having a lower water content. The more water that you have, the more that increased temperatures can cause denaturation of molecules like proteins. There's got to be some water present for denaturation of proteins to occur. Well, that's one of the threats that bacterial cells oftentimes face that can lead to sporulation is the environment dries up. That would seem to be a risky thing for the cell. But oftentimes, one of the things that causes the environment to dry out is an increase in environmental temperature. What does increased temperature typically cause? Denaturation of proteins. But in this low water environment, even at relatively high temperatures, proteins don't denature as much. And so this is a protective mechanism. In the cytoplasm, there are also a group of proteins called SAPs. SAP stands for small acid-soluble proteins. And these proteins seem to have a number of protective functions, particularly for the DNA, to prevent it from being damaged. From things like radiation, for example. Radiation is another thing that can trigger formation of endospores. If you do some chemical analysis of the cortex of the spore, the layer that surrounds the core, one of the things that you'll see is that there's a very high concentration of calcium ions in the cortex. There's also a high concentration of a substance called dipicolinic acid. Don't confuse that with diaminopimelic acid. Diaminopimelic acid is one of the unusual amino acids that we found in that inner bridge region of peptidoglycan. So this is something different. I'm not going to get into the chemistry of it, but these two particular compounds interacting with one another really help to contribute to that desiccation of the core and water removal from that core cytoplasm. A lot of times people make the analogy that the core cytoplasm is really pretty much like a gel. It is so dehydrated and has such a low water content. So this particular slide illustrates what can happen in the process of sporulation and how a portion of the cell's chromosome can be separated off following nuclear division. And the mesosome can play a role in segmenting off this portion of the cytoplasm, forming this complex, dehydrated, multi-layered structure, the endospore. That vegetative cell, the sporangium, may die and release the spore to the environment. It may not. But if environmental conditions that initiated endospore formation are reversed, then the spore can germinate and in a couple of hours give rise to a new vegetative cell. One of the things that tends to happen in germination is that enzymes become activated in this cortex wall that break the cortex wall and allow water to move back into the cytoplasm of the cell and cytoplasm of the spore, and kind of reactivate metabolism, and eventually we have a new cell that emerges from that particular resistant structure. So pretty nifty adaptation. The existence of endospores has some implications. One is that being resistant to increased temperatures, things that will normally kill vegetative cells like boiling water are not effective in killing endospores. In order to kill the endospore as well, we need water to get up to a higher than normal boiling, 212 degrees Celsius. And the only way we can do that is by increasing the pressure to elevate the boiling point. Therefore, autoclaving is really the only way to effectively destroy endospores. Boiling water alone won't do it. So that's why we autoclave things, so that we not only kill the vegetative cells that can be contaminants, but also kill the endospores, which could give rise to new vegetative cells. Another cytoplasmic set of structures that we can find in some bacterial cells are inclusions and granules. The distinction between inclusion and granules is that inclusions tend to be organic molecules, and oftentimes they are surrounded by a piece of membrane. Typically that membrane is plasma membrane. These are not considered to be organelles. Granules are inorganic materials, no membrane surrounding them. Basically, these represent strategies for storage of material within the cytoplasm. Things that the cell might need later. Things that maybe the cell produces as a result of its metabolism that it might use later. We're going to do some tests for both inclusions and granules, and this will be additional information that you can use to try to figure out what your unknown is. Does it have this? Does it have this? Does it have this? One of the tests that we will definitely do is a test for a PHB, sometimes called a fat inclusion. PHB stands for polybeta hydroxybutyric acid. It's a fat, okay? Butyric acid is a fat. Good strategy, potentially, to store some fat in the cytoplasm of your cell? Sure. Good source of carbon, good source of nitrogen. Cell starts to run out of food resources. You can then potentially use those fat molecules that are stored in its cytoplasm. This is a representation of a cell containing numerous PHB granules. The red things right here are the PHB granules. This particular stain uses a stain called Sudan 3. Sudan 3 is a commonly used fat stain. You've actually had experience with Sudan 3. If you remember back to cell biology when we did the biological molecules lab, and sitting at the end of the tables down here were some bowls of a red dye. And you spotted over there probably some materials on that circular filter paper, some fat, some cream, some oil. Remember doing that? And then dipping it in that Sudan 3 dye. The Sudan 3 binds the fat molecules. And then you just rinse it off with water, let it dry, and if you had red spots on there, that indicated fat presence. Well, that's basically what this particular test is doing. It's looking for these molecules of fat in the cytoplasm of the cell that bind the Sudan 3. So we'll do a PHB stain. Another granule that some bacteria produce are granules of glycogen. Remember, glycogen is basically sometimes called animal starch, polymers of glucose. What would be a good, easy test we could do to test an unknown for glycogen granules? And glycogen is similar to starch structurally. Iodine test, okay? Remember, iodine binds with starch to form that blue-black color. Does the same thing with glycogen as well. Some granules that can form within the cytoplasm of the cell include polyphosphate granules. Sometimes bacterial cells will store up phosphate for later use. Need some phosphate to make nucleotides, for example. So you can make your DNA and your RNA. Phosphate granules are also known as volutin granules, also known as metachromatic granules. We used to do the volutin or metachromatic granule stain in lab, but it's kind of like the flagella stain. It's really tricky, doesn't work very well. And so this is one of the ones that we kind of abandoned because it really didn't give us that much useful information. Another type of granule that can develop within the cytoplasm of the cell are sulfur granules. When we talk about bacterial metabolism, we'll talk about the fact that some species of bacteria are able to carry out a process called anoxygenic photosynthesis. When you put "an" in front of something, what does that mean? What does anaerobic mean? Without air, okay? Anoxygenic photosynthesis, photosynthesis that doesn't produce oxygen. What's that? Well, in anoxygenic photosynthesis, one of the things that can happen is that a gas, hydrogen sulfide, not H2O, but hydrogen sulfide, can be used in the photosynthetic process, and instead of producing O2, can produce sulfur granules. And those sulfur granules can accumulate in the cytoplasm of these photosynthetic bacteria. Another inorganic molecule that is sometimes stored in the cytoplasm of the cell are iron granules. Here's a bacterial cell And it's got all these little iron granules. It's hard to see it right here, but they're in black right here. The ones that are labeled MP. And they're all lined up on one side of the cell. Those iron granules are also called magnetosomes. And they allow bacteria to align themselves in magnetic fields. Interesting response, interesting behavior. It's like they have a bunch of little magnets in their cytoplasm. Well, in addition to these endospores and granules and inclusions, all bacterial cells have to have ribosomes. So that's not going to be differential for us. Remember that ribosomes in prokaryotic cells are the 70S variety, made up of the 50S and 30S subunits. So that isn't that instructive, although analysis of the ribosomal RNAs that make up these particular subunits, and particularly sequencing the genes for those ribosomal RNAs, has been very instructive. It was ribosomal RNA sequencing that was really the basis of this new system of classification, the 3-domain system of classification. We'll come back and talk about 70S ribosomes versus 80S ribosomes when we talk about antibiotics and antibiotic resistance particularly. Additionally, bacterial cells must have nucleic acid, DNA and RNA. The DNA is typically highly clustered and coiled into a central region of the cytoplasm, but it is not membrane bound, so it's not a nucleus. So the genetic material is referred to as the nucleoid. This is a bacterial cell that has been lysed. It's probably an E. coli. And all of this represents the bacterial's genetic information that has been released when that cell lyses. There's a lot of it there, isn't there? And the only way you can cram all of that into this small cytoplasm is to highly fold and supercoil that DNA. So it's very twisted up. It's in a very compact state. And that supercoiling is essential for a cell to be able to survive. It's got to be able to package up its DNA in a supercoiled fashion. There is a antibacterial drug called Cipro. Some of you may have heard of it before. That interferes with DNA supercoiling and consequently is antibacterial. Cipro is one of the antibiotics. It oftentimes is used for treatment of traveler's diarrhea, for example. It is also the antibiotic of choice for treating anthrax. Far fewer genes in the typical prokaryotic cell than in the eukaryotic cell. Typically about 3,500 genes in a typical bacterium. Numbers vary tremendously, but that's a good range. At any one given time, only about 1,000 of those 3,500 genes are being expressed. So there's a lot of regulation of gene expression that's taking place. Remember back to both cell biology and genetics. The operon model describes one of the ways in which regulation of gene expression can occur in prokaryotic cells, a way in which bacteria can adapt to environments, like all of a sudden lactose is available, so the beta-galactosidase gene is induced by the lac operon. A lot of turning on and turning off genes occurs during the lifetime of a bacterial cell. We're not going to really get into that. That's really a province of genetics, and so I leave that to Dr. Morgan McCombs. One last thing that I want to mention that we're now appreciating exists within the cytoplasm of virtually all bacterial cells are proteins that are organized into little tubular structures called actin filaments. This can form somewhat of a primitive bacterial cytoskeleton. And that's a term that 10 years ago, when I was teaching this material, I would have never even referred to, a bacterial cytoskeleton. Until microscopy had improved, we pretty much assumed that a cytoskeleton was just something that you didn't find in bacterial cells. But now it appears that these actin molecules can form primitive tubular structures, which might be the forerunner of a cytoskeleton structure in eukaryotic cells. Even some speculation that these actin filaments might play some role in separation of the chromosome following duplication when bacterial cells undergo binary fission. Maybe this and the mesosome cooperate together to play the role of separating those duplicated chromosomes into the two halves of the cell that's dividing. So we're always finding out something new about bacterial cells, particularly as technology improves and microscopy improves. So that's the overview of the bacterial cell structure. So we will start with bacterial classification. I'll make sure those PowerPoints are posted as soon as I get back from class now. I'll also post the lecture notes for this particular section. And I'm also going to send out an email today about making medicines. It's not quite available yet. We think it'll be available this week. There's an element of making medicines that will be required for the course. There's an element that will give you a lot of optional points if you choose to use it. And then also there is an opportunity for anybody that is a senior this year in this class to combine completion of making medicines with a potential Lilly internship. And so I'll send out that information today. Okay? All right. I'm sure you're behind in a lot of things, and so
No, I'm actually not.
Aren't you?
It's not that bad.
Okay. So what works for you in terms of? I mean, basically, I'm fine with you reading through the material on making media. I don't see any reason to have you
Okay.
pour the stuff together, but I would like you to read through and see what you have to add.
And that was the lab from last week.
That was the lab from last week, and also the microscopy. So we're going to have to work on
So is
microscopy.
that the first two labs then?
Yes.
Or
The first lab was microscopy and making media. The
I
lab
read
Friday
through
was
that
the aseptic technique. What?
Okay. I read through the whole first one
Okay.
the other day, so I haven't read through the aseptic one then.
Why don't we maybe do? Well, we should probably try to do the aseptic. We should probably try to do the aseptic as soon as we can because you're going to need some of those techniques in lab on Wednesday.
Okay.
What's your schedule like tomorrow?
I'm done at 11 tomorrow.
You're done at 11 tomorrow?
Yes.
Okay, I have a faculty meeting. I'll tell you what, if you can, why don't you come in at 11 and work on the microscopy part
Okay.
and do what you can, and I'll show you tomorrow in class. All the slides are set up over there,
Okay.
and I'll have to get your microscope down too, so we do that. And then I will come back after the faculty meeting, and we can sit down and work through any problems you had with microscope.
Okay.
Tonight, read through the aseptic technique lab. It won't take that long.
Okay.
And after we do the microscopy, I'll walk through the aseptic technique with you.
Okay.
It won't take that long. I mean, I would guess probably if I get back a little bit after 12, whole thing shouldn't take more than an hour, maybe an hour and a half,
Okay.
and then you can be caught up.
Sounds good.
Okay?
All right. I'll be here tomorrow at 11 then.
All right,
Thank you.
sure.